Dear colleagues, as we unfortunately had to cancel this year's World Congress in Montreal, we thought it would be a good idea to at least share some highlights from the virtual Congress that took place in February. Before the video starts, I would like to announce that the 2022 World Congress in Taipei, Taiwan will take place from June 9th to the 12th. I look forward to seeing you there in person, and now please enjoy the video. Oh, and don't forget to subscribe to our YouTube channel below for free so you can watch more videos in the next weeks. Thank you.
Thank you for kind introduction. Good morning, good afternoon, or good evening, everyone in the world. I'm Noriko Osumi, professor at Tohoku University School of Medicine, and currently serving as the president of Japanese Society for Neuropsychopharmacology. One of the example, the maternal origin of the health and disease, and there was a very severe famine during the World War II in Netherlands. So, and the people who are exposed to this severe famine will develop later in the schizophrenia like this, especially those people who are exposed to the severe famine during the first trimester. So this was the model in mice. This is my the previous students and my colleague now in mid-weekend, so whose name is Motoko Maekawa. So she made this kind of the experimental paradigm. So before the mating, the female mice are fed with the fatty acid deficient diet, and then crossed with male mice, and then the baby, the offspring are born. And so we would like to ask that what kind of phenotype those offspring shows. So very typically, so these mice shows a hypersensitivity for NMDA receptor inhibitor, which is a good model for the psychosis. And actually, so there is a hyperactivity on the brain in case of these fatty acid deficient the mice. And also she found that the reduction of the expression in oligodendrocytes related genes, you know that oligodendrocytes are so much rich in lipid. So, and another example has been done by another student, the Nobuyuki Sakayori, now in Hiroshima University. So the Nobu treated mice with a modern diet, which means that the more the seed oil, which means that N6, the fatty acid rich, but poor in I'm sorry, N3, the diet, the fatty acid. So this is the recapture of the modern diet, which is very rich in the seed oil, but lacking the fish oil. So the outcome of this, the diet, the exposure is that this is a normal development in the neurogenesis is followed by the gliogenesis. But in case of the mice, the exposed in utero that impaired the imbalance the fatty acid, those shows a shortened period of the neurogenesis, and the precocious the gliogenesis is occurring. By that way, the production of the neurons are reduced, and which is actually confirmed by using the neurostem culture system and exposed with the metabolites of these fatty acid of the N6 and N3. So you see that the more the neurogenesis occurred using the fatty acid metabolites of the N3 type, but while these are the N6 derived the fatty acid metabolites increase the number of the glia cells, in this case, the astrocytes. So, and how about the behavior? So actually, so these the mice shows increased anxiety in case of the open field test, as well as the elevated maze test like this. So, and then, so here I just would like to a little bit more emphasize about our ultrasonic vocalization paradigm, because that we would like to know that individual difference. So the maternal separation induced PUP ultrasonic vocalization can be considered as an infant crying, and so which can be tested in a very early postnatal stage like a P3. And so I think, so because that we can collect a large number of the data, so this is very suitable for the mathematic analysis later. So my talented student Lindy Mai analyzed these the behavior, the ultrasonic vocalization. So the recorded ultrasonic vocalization can be switched to the image of the data like this. And this image data can be further analyzed using the machine learning type of the analysis like VAE modeling approach, the variation autoencoder. So first, we'd like to know the difference of the young father-derived group and the aged father-derived group. And as we notice that, so there is a less number of the syllable and less duration and impaired interval the development like this. So these are the further detailed analysis of the previous one I shown before. You know that what's happening in the epigenetical level, because that there is a two paper coming out in Nature in 2011 to 12 that there are of course the increase the de novo mutation in the aged father, but later for example, in 2016, the Nature Genetics paper showed that the contribution of this de novo mutation is not so much the dominant in the offspring. So another example is also coming up in the 2019 in the Nature Communication as well. So I would like to ask the epigenetical mechanism derived from the father. So which means that's a sperm. And there is a longitudinal cohort study in the United States by the contribution of the families who has autistic kids. So the one paper is coming out in 2015 that the paternal sperm DNA methylation can be associated with autism in the offspring. And also in this paper, they show that DNA methylation is also commonly observed in the postmortem brain of the ASD patient. So we wondered what's happening in the mouse, the sperm in the aged one. So we collaborated with Dr. Kono, Professor Kono in the Tokyo Agricultural University at that time. So he is an expert in the whole genome methylation analysis using the second generation sequencer. So we have came up with the aged father derived aged sperm shows 16 hypermethylated regions, while more large number 96 hypomethylated region, which is typical in the aged sperm. And we analyzed these the hypomethylated regions, and are there any the geoterm related to the nervous system? And the answer is yes, in case of the human ontology and also the mouse ontology, that these the hypomethylated region and enriched with GO terms with neurons or nervous system or neural function, etc., etc. And also we have came up with a common motif, which is a binding site for the NRSF or REST, which is also coming up with the chip atlas, which is established by Dr. Oki in the Kyushu and now in the Kyoto University. So in these the hypomethylated regions, 37 regions commonly showing this REST binding motif. And also in our laboratory, we are also asking that the epigenetic mechanism focusing on the spermatogenesis, and especially asking what kind of histone modification can be altered. So some of you have known that the histone can be replaced with the protamine, but the recent data show that nearly 15% of the histone can be remained in the fertilized egg. So we wondered what's happening in the sperm during the spermatogenesis, and we have checked these kind of activated related the histone modification and also inhibition related one as well. So this was done by my talented the student Misako Tatehana, and she carefully examined the immunohistochemistry of the testis and asked the semi-quantitative analysis using these different the histone modifications. And so these are very complicated data and summarized like this way. And I just would like to show you this summary the table. And there's lots of up and down of the inhibition or activation of the histone markers in a very different way due to the aging of the male mice. But interestingly, so some of these the difference in the spermatogenesis can be also last in the sperm as well. This is the Western blot analysis, and these two histone modification is actually increased in the sperm as well. And we have patented this one, especially H3K79 trimethylation. So this intensity is negatively correlated with the number of the USV of the offspring derived from this the father, from which the sperm samples is taken like this. So in future, we would like to ask that are there any kind of some drug can be developed by checking the alteration of these the histone markers. So we'd like to develop some anti-aging epi-drug in future. So I'd like to finish my talk by summarizing that both the eggs and also the sperm is very, very important, and which can effect of those effects can be last for the next generation as well.